Okay, so now for the next talk, we move from the Swiss Parliament to actually the human brain. And with that, I would like to introduce our next speaker, Francesco. Francesco is a section manager and is responsible for leading the machine learning team at the Blue Brain project. He leads the efforts to develop machine learning tools in order to improve and accelerate the neuroscientific workflows of the Blue Brain project. So his background is in applied mathematics and has previously also worked in the field of aerospace. So welcome, Francesco.
Hello everyone, my name is Francesco Casalegno, and as you heard, I work as section manager and machine learning engineer at the Blue Brain project. So the Blue Brain project is part of EPFL and it focuses on computational neuroscience. And what I would like to share with you today are some of the lessons that I learned while working and collaborating together with the neuroscientists during this project. So first of all, let me give you a couple of words of introduction to what the Blue Brain project is doing and to what our machine learning team focuses specifically. So the Blue Brain project is working in this beautiful campus that you can see in the picture, and its mission is to reconstruct and simulate the mammalian brain, more specifically starting from the mouse brain. And as you may imagine, this is an extremely challenging goal. So we're talking about more than 70 million neurons, and it is estimated that the number of synapses in the mouse brain could be around of the order of magnitude of 100 billion of synapses. So we're also working with very diverse teams. And certainly, since we're talking about reconstructing mouse brain, we have quite a lot of neuroscientists, but there is also an equal number of IT specialists who are building software tools and maintaining the infrastructure and the hardware and the supercomputer that is necessary in order to run these neuroscientific simulations. And the way the Blue Brain project is working is through this iterative workflow. So we're getting the data, we're building the models based on the data that we get, we run a simulation, and then we try to refine the models based on the results we obtain. So in all of this process, all of this workflow, the data plays a key role. Since data, we can obtain it from experiments, from public databases, from inference, from data that we already know, and so on. And so this is where the machine learning team really enters the game. And our goal is to try to support the Blue Brain project scientists by creating various machine learning-based tools so that they can try to have solutions implemented and designed by us and that they can then use in their everyday work. And so I think that there are three main directions where machine learning can help the work of the neuroscientists at Blue Brain. So first of all, we can accelerate workflows, which otherwise would require human labor and be extremely tedious and long. Second, ensure reproducibility and consistency across different runs. And finally, enabling the creation of automated end-to-end scientific pipelines. So now that I've introduced a bit what Blue Brain and our team does, I will also like to give you two examples of concrete projects that we have worked on. First project is called Atlas Alignment, and its mission is to try to merge different brain atlases. So in particular, we have access to what is called the reference brain atlas, which means that they took a mouse of a brain, a physical mouse brain, and they sliced it, and to each of the slices they applied a particular kind of staining, which is called Nissl staining. And then they also took various other kinds of mice, and they applied various other kinds of staining. So they're called in situ hybridization, and they basically show you different gene expressions. So the problem with that is that the staining procedure generates all sorts of tears and bubbles and irregular deformations to the slice. So what we would like to do is to be able to take these gene expressions and map it onto the reference atlas. So to create this deformation, this alignment, or to talk about machine learning terms, we're talking about registration. So what we want is to try to predict the deformation that aligns the gene expression onto the reference atlas. And to solve this problem, we used a neural network that first tries to create an affine transformation, so it's called global module, and then it's followed by a local module which predicts a more like pixel-wise deformation. And by the way, all of our projects are open sourced and published, so here you have the links in case you want to find more about it. Second project, which is actually surprisingly similar to what you heard in the previous talk, is about literature search and mining. So in this specific case, we worked on the COVID-19 So you may wonder how this is related to the Blue Brain core mission. And the answer is that in 2020, basically, the world almost stopped working. And we really wanted to try to put our energies and our knowledge at the service of the community. And in particular, we found out that Core 19 was a massive open literature database that was published with more than 600,000 articles, including 250,000 full text. And of course, a single human would never be able to read all of these papers and extract knowledge from it, which is the reason why we implemented machine learning models in order to make this huge literature database searchable. So first of all, we had to train a custom machine learning model for searching the COVID literature. And we used BioBERT as a basis. But if you're maybe wondering, why did we have to train our own model? Well, BioBERT was actually published in the summer of 2019. You may remember that what happened in December 2019 was the arrival of COVID. So all of this literature is all about the SARS-CoV-2, coronavirus disease, and so on, and these are all terms that don't exist in BioBERT. So we had to fine-tune it on our own literature. And then we extracted various entities with named entity recognition. We linked them to the corresponding nodes into our ontology. And from that, we created a knowledge graph that neuroscientists can then, and biologists could then explore and navigate in order to extract knowledge and information. All right, so now that I've talked to you about what we're doing, I would also like to share with you some of the challenges that we have found with our team. So first of all, one of the issues that we have encountered is that when you're talking about supervised machine learning tasks, the common assumption is that you're given X and Y, so there are your inputs and target outputs, and Y during the training and the evaluation is assumed to be the ground truth. Except that in our case, it's not the ground truth. It's something slightly different that I would call gold standard, and it comes from human annotation. So what is the problem with that? Human annotations can be noisy because humans can, of course, make mistakes, or at the very least, they can be subjective, which means that if you ask two different humans to annotate the same thing, they will give you two different answers. So for instance, here I have, again, the example of atlas alignment, and we have this reference atlas and the input, which is this gene expression that we would like to align to the original reference. So what we can have as ground truth labels, for instance, if we want to create a machine learning model, it could be, for instance, the kernel section number, so we know exactly the section number, because this was something that was recorded in the metadata, and this is information which is known. Or, for instance, the gene expression that was used to stain the input. And, for instance, we know that this was used NPY gene. So this is objective ground truth. It's not something that we can question. But we're working with something different. We're working with gold standards. And two examples of this are the deformation that aligns the input with respect to the reference. And if you ask two humans or even the same human, they may give you slightly different answers. And for instance, another case is brain subregion segmentation. So telling for each pixel which subregion it corresponds to. So why does it matter? First point is garbage in, garbage out. So if you train your model with labels which are noisy and contain errors, your model is also going to infer things which are wrong. And second problem is biased evaluation. So you also want to see how good your model is doing. But if you're validating your model against a data set which contain errors, you're also going to have some troubles. Also related with the data annotation and with the fact that humans do not always agree with each other, and in fact not always with themselves sometimes, is the problem of inter-rater agreement. So this is sometimes you go to the doctor, you don't like the answer they give you, and you go to a different doctor who is going to give you a different answer. So both of them are very well-respected professionals, except that they may slightly disagree on certain annotations. So I will give you a concrete example. So here again, we have our reference atlas and an input staining. And we asked annotator number 1, an expert in the field, to predict the alignment. And they give us this slice. I think arguably it matches very well the reference. Except that then we asked expert number 2, and they gave us a slice, which if you have good sight, you may recognize is not exactly the same. It's very similar, but it's not the same. And so why does it matter? Number 1 is setting expectation, which is something that was also mentioned in the introduction to this track by Tarun. So when we start the project, should we aim at reaching 100% accuracy? And what does it even matter? Like accuracy with respect to what? because accuracy with respect to expert 1 is not the same thing of agreeing 100% with expert 2. And also definition of done, because these projects sometimes, especially in academia, they don't really have a clear-cut stopping criteria, so you may be working for years trying to keep improving your model, and you never know when you should stop. And also finally, training and evaluation. Should you train or evaluate with labels of expert 1 or expert 2? Another problem related to annotations is that they are expensive. So expensive here, I put the nice emoji of flying money, but you could also think of that as time, or even just convincing the neuroscientist who asks you, well, but can't your model learn by itself? Can it do like some sort of unsupervised learning? And sure, there is like huge field of self-supervised learning and unsupervised learning, but sometimes you need to convince them to give you the annotations. And this may be expensive, time-consuming, or even just in terms of human relationship, hard to convince people to give you more data. So let me give you a concrete example. So we have the Monopoly Man, which give us a certain allocation in terms of time of money, and we get 200 annotated samples. We reach 80% accuracy with our model. Okay, so maybe the management is very happy with these results and says, okay, so I give you 3 times more money or time, and now you have 600 annotated samples. Which accuracy do you think that you can get? Which is also related to the overwhelming question, how many annotations are you going to need for your model to train it? And which samples do you want me to annotate? Which is a question that scientists always ask us. And why does it matter? Well, of course, we want to optimize cost and time allocation in order to optimize the return of our investment. And finally, something that is more specifically related to science, and is something that I haven't heard that much actually here during these days, because I think it's something that we care more in science than the industry. Industry is more about MLOps, and for us actually it's slightly different. So tracked and reproducible science, being able to reproduce the same results is something that is very relevant to us. So usually the way it works is that we have a machine learning pipeline. So we ingest some data, we have code and environment, so different operating system, different Python runtimes, different library versions, and so on. And then we generate trained machine learning models and validation metrics. So this effectively translates in machine learning artifacts. So I don't know if this rings a bell to any one of you, having results XGBoost, results XGBoost 2, results best, results better, these kind of things. And it's really hard then if they ask you, can you just reproduce the experiment for results best? And this is a bit hard to being able to recover that. And why does it matter? Well, first of all, because the peer reviewing process, which is unfortunately something that is, in my opinion, not working that well yet for machine learning, is that the paper readers and the peer reviewers may want to rerun the experiments, and they would maybe want to see the same results that you're obtaining, because otherwise they may question your results. And I would even say that this is the very fundamental of scientific method, so being able to reproduce experiments and verify the results. And finally, also in terms of deployment, we want to ensure the results will be consistent for future users and that something will not break. All right, so here are all the challenges that we have faced, and now I would like also to try to give you some nice perspectives about solutions, or at least lessons learned and things that we have tried to counteract all of these challenges. So first of all, the distinction between gold standard and ground truth. One of the solutions that we can try to apply is to use robust fitting methods. So in particular for regression, we have RunSuck, which is a model which will try to iteratively try different subsamples of the training set and fitting your model on these samples. And then try to see on out-of-sample prediction how far off the predictions are with respect to the supposed gold standard. And if many of these samples appear to be outside of the threshold, maybe this model is not considered, and then instead they took the model which fits best and has the most inliers. And all of the elements which are outside of this threshold are not actually used for the fitting of the model, which is also very convenient because it allows you to spot potential outliers, which means samples which have wrong labels white true, and the model is not impacted by them. So this is about regression. And about classification, we have this other tool, which is called CleanLab, implementing a novel machine learning framework called confident learning. So here the idea is that we can never access the ground truth labels. We only have access to noisy labels. And the idea is based on out-of-sample estimates, out-of-sample predictions, so try to estimate which samples may be noisy, and then train the model on the samples which are not considered to be noisy. And I think that, so this picture is taken from the CleanLab paper, and I think it's quite interesting because it's shedding some light also on modern machine learning benchmarks and standards, such as the ImageNet dataset. So they found out that, for instance, this sample has the ground truth annotation, well, supposedly ground truth, Patas monkey, but it clearly is not a monkey without being expert in animals. And why this is relevant is because we are evaluating constantly. We're trying to achieve state of the art with respect to certain annotations, which are actually containing some noise. Second thing was about the inter-rater agreement. And so here, sometimes we can compute specific test statistics. So for instance, if you have 2 or 3 evaluators and they have ordinal or categorical annotations, you may be using Cohen's kappa. Or if you're working with more continuous values, you can use various kinds of correlation matrices. And why these test statistics are nice is because they typically include chance correction. So for instance, here you have the formula for Cohen's kappa, which does not simply measure the accuracy, that is like the brute percentage of agreement between 2 different annotators, but also corrects by the factor of what if like they just agree by chance, because 80% of the samples are anyway true. And another choice is, for instance, to use a symmetric evaluation metrics. So for instance, you could use intersection over union, or accuracy, or perceptual similarity, anything that you want that can be symmetric. Why symmetric? Because, for instance, if you used precision of annotator 1 with respect to annotator 2, is not the same thing of annotator 2 with respect to annotator 1. And then what do we do once we have computed this inter-rater agreement is that we usually tried to draw a line and to tell our users from the very beginning, look, like this is the best accuracy that we will ever be able to achieve. And not because we're not good enough and we cannot achieve 100% accuracy, but rather because this is the intrinsic uncertainty contained in the annotations. And so this is an important baseline that we try to draw. And related to this, I think that other important baselines are, first of all, before trying to recover some sort of a complex machine learning model, you should also maybe try some non-machine learning model, first baseline, and also second baseline, maybe try also some naive machine learning model. So that maybe your problem is already solved there, and even if it's not, then you can flex how good your model is with respect to non-machine learning. Concerning the fact that annotations are expensive, and we have a few of them, so here there are 3 strategies that one can think of. First off is transfer learning. So for instance, when we were talking about searching the literature, the nice thing there is that you have many ways to train natural language processing models which are self-supervised. And this can also offer you like an interesting pre-training, and then you can perform then a fine-tuning on a given task, such as named entity recognition. A second thing that we can also try to think about is active learning. So we can try to pick the examples where the model feels less confident about and ask the annotators to annotate those first, so that we're going to reduce the uncertainty as much as we can. And finally, there is also another strategy, such as indirect feedback. So once the model is already producing some nice predictions, we can just ask the annotator to say yes or no, like this annotation is correct or this annotation is wrong. And of course, we reduce the signal, the information that we have in these annotations, but it's maybe faster from the annotation perspective. And I also wanted to mention this library, which we found very nice. It's called Prodigy, and is done by the same developers of the Spacey library, which is a staple for all NLP projects. And it's very nice. It implements, for instance, active learning and indirect feedback. And another thing that we also found interesting is to plot this accuracy versus training set size curve. So you remember the overwhelming question that users ask you all the time when you start a machine learning project, which is, how many labels are you going to need from me before your model starts to work in a decent way? And typically this follows a power law. And yeah, so this means also like diminishing returns. At some point, you may not want to ask them to label 1 billion sentences to improve by a margin, which is maybe irrelevant. And then finally, for tracked and reproducible science, two things that we have employed with success are, on the one hand, Docker, which tried to solve the problem, but I promised that yesterday it worked on my laptop. And then you ship the code to someone else and it doesn't work. So here is very nice because you can package your machine learning application together with the operating system, the libraries, the binaries that you need, and the Python packages and so on. And something that I also found sometimes in this conference, which is very exciting to see also other people using, is this tool called Data Version Control, DVC. It has an interface which is very similar to Git, but works similar to Makefile. So, which means that if you have some of the steps in your machine learning pipeline which have changed, you're able to rerun the experiment, starting only from the stages of the directed acyclic graph representing your machine learning pipeline, which are actually needed. And what this DVC is really good at is versioning data on top of machine learning models. Also related, MLflow is also another nice library. Okay, so thank you very much for your attention, and please feel free to reach out in case you're interested in discussing anything with me.
Thanks a lot, Francesco, for this insightful talk. It was very nice, and I think we have a lot to discuss later. So you pointed out some very nice points. So do we have some questions in the audience? Okay. Thank you.
Thank you for a very nice talk. I just had a question on your last point on traceability and reproducibility. I was just wondering if you had considered, or if you're aware of the fact that Renku would, take care of a number of these things, if you considered it. I'm just curious because basically Renku would provide an entire workflow control version system. It is built on Docker, and it will do, among others, the data version control that you're mentioning. So I'm just curious if you considered it or not.
I'll be honest with you, it's something a bit new to me, so I will definitely have to look more into that. We started using DVC years ago because it was already pretty popular, but of course we're open to alternative solutions.
Thanks.
Do we have any other question?
So thank you again for the interesting talk. I was thinking about, you were talking about this inter and intra operator variability. Is it really big? So let's say if you would represent the same picture to the same person, are they gonna do kind of like a very similar annotation, or is it not that important, like the person effect?
So I would say that it really depends on the specific task. So if we're talking about the one about aligning two slices one on top of the other is a particularly challenging task, even for a human. And there is a lot of room for interpretability. So there, I think that the inter-rater agreement could actually be significantly impacted by this. But on other projects, to mention one, we're classifying neurons based on their morphology, so M-type, morphology type classification. And there we also wanted to assess the intra-rater agreement, but we had the problem that one of the annotators has a visual memory and remembered from 5 years before, and could actually tell the name of the cell, say, oh yeah, I remember that cell. That's of course TPCB class. And so there the intra-rater agreement is 100% because this specific person remembered the cell by heart.
Okay, thank you.
So Francesco, thanks again for this very nice talk. Thank you very much.